We are continuing our discussion about carbohydrate derivatives. In the earlier video, we discussed elaborately about various amino sugars. In this video, we will discuss about deoxy sugars. Deoxy sugars, which means one of the hydroxyl group will not have the oxygen. It is removed. There are three classic examples of deoxy sugars which are found in nature. For example, the first one is beta deoxyribose. So this is the first one which is present in the DNA molecules. So you can make out that at the position number 1, this is position 1, 2, 3, 4 and 5. At position number 1, an oxygen is removed. Look at the next deoxy sugar, that is alpha L rhamnose. So at position number 6, it is deoxy. Oxygen has been removed. Similarly, L-fucose, which is found in algae, is also found in blood group substances. At position number 6, the oxygen has been removed. Deoxy sugars play a very significant role in living system. We know that deoxyribose is present in our DNA structure. And presence of the deoxyribose makes the DNA highly stable. So it has got a hydroxyl group at the third position, and this third position makes it possible for forming either a backbone of DNA molecule. What happens if it were a ribose sugar instead of a deoxyribose sugar? Ribose sugar is found in the structure of RNA molecules. If it were a ribose sugar, generally what happens is in alkaline condition. So this we should understand that there is a phosphate group attached. In the case of phosphodiester bond, which forms the basic backbone of DNA structure. So in alkaline condition, what will happen is, if there is a hydroxyl group here, this phosphate group binds with this. So in other words, this will form a phosphate ester. It forms a kind of a phosphate ester here with the second and the third carbon atom. So therefore, this is known as 2,3 monocyclic compound. Because of the formation of the monocyclic compound, this structure can be very easily broken. This is one of the reasons why DNA will not have deoxyribose, whereas RNA, which is not very stable, can have a ribose sugar. We will also find many viruses using RNA as the genetic material. But then we should understand that this genetic material in viruses is compact, very well packed, devoid of watery environment. That is how the genetic material in viruses will be protected. In the next carbohydrate derivative which we are going to look at is sugar phosphates. Sugar phosphates are playing a very fundamental role in living system. We are looking at sugar phosphates especially with the metabolic functions. In most of the metabolic functions, they transfer energy in the form of phosphate. They are esterified, and this is one way by which energy is preserved, or it is transferred from one molecule to the next molecule in living system. Several examples can be given. You have dihydroxyacetone phosphate, you have glyceraldehyde 3-phosphate or glucose 1-phosphate, glucose 6-phosphate. These molecules are very, very significant in metabolism. They also play a key role as a structural component. So as a structural component in the cell wall, phosphate groups are added. Sugar phosphates play a key role. They also play a role as instructional molecules. So we know that in RNA, in DNA, sugar phosphates are playing a very important function. They also have a storage function. So there are several sugar phosphates which will be storing energy. For example, we have a phosphoenol pyruvate. It's an excellent example for storage functions. They also have a role as part of coenzymes. Several coenzymes will have sugar phosphates. FAD, FMN, NADP, coenzyme A, all of them will have sugar phosphates playing a role as coenzymes. So when you look at sugar phosphates, they are so significant in living system. Here I have kept selected certain molecules in order to highlight that the amount of energy in terms of kilojoules per mole present inherent in these molecules. And at the same time, very important to observe that all these sugar phosphates, they will have more than one pKa value. So, for example, in the pKa values of the sugar phosphate, generally it is between 1 to 2, and the pKa 2 will be generally between 6 to 7. The last sugar phosphates we will be looking today will be phytic acid. So phytic acid is generally present in plant system, and we require an enzyme known as phytase enzyme in order to degrade phytic acid. So phytic acid is, you can make out, there are 6 phosphate groups. It is also known as inositol 6-phosphate. So the amount of phytic acid can be minimized by soaking in water, boiling, germinating seeds. The amount of phytic acid can be reduced. Certain creatures are not able to digest phytic acid. For example, swine, fowl, fish, they are unable to digest phytic acid. If food containing phytic acid is given to them, they will excrete a large quantity of phosphate. Now this phosphate, as we know, it can lead to uncontrolled growth of algae, and that can lead to eutrophication. This video will not be complete unless we say a few lines about sugar acids. This is a sugar acid. This is glucuronic acid. Glucuronic acid. Glucuronic acid is one of the intermediate that is formed as part of pentose phosphate pathway. So similarly, many other sugar acids are found in nature, and sugar acids play a very vital role. Especially this will be able to give a charge to several biomolecules. Galactose can be converted to its alcoholic form. That alcoholic form is galactitol. As we become old, if there is excessive consumption of galactose, that is in the form of milk, there is a possibility that galactitol can get accumulated, especially in the eyes. So galactose will be converted to galactitol by an aldose reductase enzyme. If galactitol is getting accumulated in the eyes, this is an osmotically active substance. And this substance will draw water to itself, that can lead towards formation of cataract in the old age. This session will not be complete unless we discuss something about the common abbreviations that are used for sugar derivatives. Some of the sugar derivatives are given here for students to get an understanding about how these common short forms, abbreviations are used. Gluconic acid, for example, this is how it will be written. So therefore, all these sugar derivatives you will find in the short form that are generally present in a biochemistry textbook. I'm sure that you have enjoyed looking at these videos about carbohydrate derivatives. Keep subscribing and keep enjoying these videos.